Hello, my name is Matt Holt. I am a senior scientist in the computational biology group at PacBio, and today I want to talk to you about our tool StarPhase and how we use it for long-read pharmacogenomic diplotyping. First, before we get into StarPhase, what is pharmacogenomic diplotyping? Pharmacogenomics itself is the study of how genes influence our response to medications and other forms of treatment. Specifically, things like dosage, the rate of metabolism, and whether or not a drug is toxic are all things that can be influenced by genes. Because of this, pharmacogenomics is really broadly applicable to pretty much everyone. And in fact, at some point in your life, you will likely receive a pharmacogenomic test for the purpose of determining what treatments are best for you as an individual. That test will form a process called diplotyping, which is where we actually identify which gene forms are present in that individual. PGX diplotyping has a wide spectrum of complexity depending on which gene you're looking at. The simplest PGX genes require only the detection of specific variants or single variants that we can then use to establish which form is present. However, most genes are more complicated, requiring the detection of multiple variants, phasing them onto a single haplotype, which we can then report as a quote-unquote star allele. The majority of our pharmacogenes use this multivariant representation to represent the different forms. However, some genes, specifically the HLA genes, require even more detail. For those genes, you need to detect the full DNA sequence end-to-end for the gene, which can then be translated into the HLA nomenclature, which is depicted up here in the top right. This nomenclature defines HLA genes by their protein, cDNA, and DNA sequence in the star allele itself. Lastly, CYP2D6 is probably the most complicated of the pharmacogenes. For CYP2D6, we need to detect the full DNA sequence, but we also need to be able to detect copy number changes, specifically deletions and duplications, both of which are fairly common with CYP2D6. This makes it very challenging to actually implement pharmacogenomic diplotyping because each gene is very different from the others, and you need multiple specialized callers in order to accurately diplotype them. You may be asking yourself, why would you want to use long-read sequencing for pharmacogenomics? If we take a look at previous technologies that are frequently used for pharmacogenomics, they each have limitations. If we take a look at MLPA, microarrays, or qPCR, each of these is pretty good at detecting known variation that we're searching for when it comes to pharmacogenomics. However, they can't put those variants onto haplotypes, allowing you to phase them into star alleles. This often means that they are inferring which star alleles are present based on the likelihood of things that have been seen in the population. Additionally, things like copy number changes can sometimes be detected, but it's often difficult to establish which duplications are present in an individual. Lastly, these point tests cannot actually detect the full sequence, which would be required for something like HLA or CYP2D6. Short-read sequencing does a little better. They can, of course, detect known variants with short-read sequencing, and you can even do a very limited amount of localized phasing. Unfortunately, this doesn't really work for pharmacogenomic genes, where the genes are usually much larger than the short-read fragments. Additionally, there is some amount of copy number detection that can be done with short-read sequencing, but it's again difficult to actually determine which copies are present when those things are detected. Lastly, read fragments from short-read sequencing are usually too short to get a full sequence for genes like HLA and CYP2D6. This is where long-read sequencing comes in and solves a lot of these issues. The read fragments for long reads are often 15,000 base pairs long, which allows you to not only phase your variants, but also get direct observations of copy number changes for CYP2D6. Of course, if you have these long fragments, we can use assembly-like algorithms to actually generate the full DNA sequences for HLA and CYP2D6 as well. With all of this in mind, long-read sequencing is really the only technology that directly captures all of the known information required to do an unambiguous pharmacogenomic diplotyping. And StarPhase is the tool that we've developed to actually process that data into pharmacogenomic diplotype calls. The most recent version of StarPhase includes 35 pharmacogenomic genes. 23 of these are relatively simple genes based on variants that have been called by a tool like DeepVariant. These variants are phased using our long-read technology, and we can then assign those to star alleles with relative ease. 11 of them, which are all HLA genes, are more complicated, requiring StarPhase to do an assembly-like algorithm to detect sequences for the entire gene. CYP2D6, of course, takes this a step further by not only requiring these sequences, but also requiring copy number change detection. Now, because the HLA genes in CYP2D6 are very complicated, StarPhase will actually generate sample-specific visualizations as well, allowing a user to come behind and inspect the result for its accuracy. StarPhase itself has 3 distinct algorithms depending on which pharmacogene we're looking at. One class of pharmacogene are the quote-unquote simple class, where basically all StarPhase does is look at the variant calls and phase information from the provided VCF file. These are then matched to star allele definitions from the databases and reported as a diplotype call for that gene. The second branch basically captures all of the HLA genes. For these, we run a full-length consensus algorithm, which generates an assembly for each haplotype, and those sequences are then matched to the IMGT HLA database, which is reported as a diplotype call in our output file. Because we have these consensus sequences, HLA also includes a BAM visualization where we realign the reads back to the consensus sequences. That way someone can come behind and inspect the result. CYP2D6 is also a special case gene. It's actually very similar to HLA from a process perspective, but there is an additional chaining step where we're basically looking for copy number changes, and if we find any, figuring out which haplotypes they go with. Similar to HLA, we will have a diplotype output call for CYP2D6, but we'll also have a BAM visualization, which I have an example of in an upcoming slide. So let's take a look at one of these custom visualizations that comes out of StarPhase. This picture is showing an example from NA20129, and we're looking at the CYP2D6 gene, which for this sample we know is a star4 times 2, which is a duplication event on one haplotype, and a star5, which is a deletion event on the other haplotype. If you look at this figure, at the bottom, you can see blue bars indicating which tracks have been assembled by StarPhase itself. And then for each of these haplotypes that has been assembled, the gray bars up top are individual read observations from long-read HiFi sequencing that have been realigned back to these haplotypes. So the first section of this figure is haplotype 1 in our example, which is star5. This haplotype is very simple because the star allele has actually been deleted. In other words, there's no copy of CYP2D6 on this haplotype, so it's relatively short. There's a deletion signature, which is the star5, followed by a spacer region, and then CYP2D7. On the right, we have a much larger allele, which is haplotype 2. On this one, you can see there are 2 copies of star4.4. And what's interesting about this depiction is you can actually see reads up above that are directly aligning to these 2 copies. And this is the direct evidence from long-read sequencing that I was talking about on a previous slide. We don't have to infer that this copy number change is actually occurring in this dataset. We can see it directly in the long reads themselves. Now, this particular example is interesting because it's often called erroneously by other technologies. It's very easy for a duplication and a deletion event such as this one to look like a simple star4 homozygous call in other technologies such as short-read sequencing. Other assays will also often get an error very similar simply because it is very difficult to detect a duplication and a deletion unless you have direct observational evidence. At this point, you may be asking yourself, how does StarPhase compare to previous datasets that have been reported? So on the left here, I have a benchmark comparing StarPhase to reported outputs for about 147 datasets. And what you can see is that the vast majority of the calls from StarPhase are matching those from these previous results, which are showing up as green in this figure. You can also see there are a handful of differences, some of which are caused by things like database updates, and some of which are true mismatches to these previous results. When we went and manually inspected the differences, we found that most of these discrepancies are actually limitations of the previous technologies. In some cases, they could not detect the phasing of certain variants, and in other cases, important information at the DNA sequence level was simply not detected by the assay. Fortunately, with HiFi long-read sequencing, we were able to observe all of these effects and verified that the answers reported by StarPhase are correct. Second, we're also able to look at large pedigrees, like the Ceph pedigree 1463, which includes NA12878, which you may know as HG1. If you look at this pedigree on the right for CYP2D6, if you were to spend some time checking the results here, you would find that there's no inheritance violations across this entire tree. We've actually done this for all of the genes that are listed on the left here for StarPhase, and so far we've not identified any inheritance violations even in these large pedigrees, giving us stronger confidence that StarPhase is in fact finding the correct results even when we have very complicated genes to work with. We've also looked at doing population studies with StarPhase. In particular, we gathered a large cohort of datasets, about 1,450 HiFi whole genome sequencing datasets, from 5 different sites. We ran StarPhase on each of these datasets, gathered the outputs, and then grouped them into ancestry in order to get population-level statistics. With this data, we were able to do a variety of interesting things. First, we could actually take our distributions and compare them to previous existing distributions that were generated by other studies. For example, in the top right figure shown here, you'll see pairs of bars for each ancestry type in our study. On the left are lighter colored bars, which is actually a distribution from the All of Us program, which we have copied into this figure for comparison. And on the right, the darker colors are those from StarPhase themselves. And what you can hopefully appreciate is that the distributions of haplotypes in our population look very similar to those from the All of Us program. For example, if you were to take a look at star 37, which is the orange bars in this figure, you'll see that they're enriched in the African, East Asian, and South Asian populations, but depleted in the Americas and European ancestries. This is for both the All of Us program and our cohort, giving us stronger confidence that StarPhase is reporting distributions that match those from previous studies. We can also use StarPhase to generate new population distributions for genes that were more complicated to do with these previous technologies. For example, CYP2D6 did not have a comparator distribution from the All of Us program, but we were able to make one using our data from StarPhase. The figure on the bottom right here shows that distribution for CYP2D6, and hopefully you can appreciate that there are some interesting phenomena that happen in this dataset. For example, star 36 plus star 10, which is shown in orange stripes, only appears to exist in the East Asian population or in the unknown population, indicating that it may be of interest to certain researchers who are studying those populations, but less interesting to other researchers. We can also use StarPhase to quantify the number of novel alleles that were detected in our population. Since we're generating full-length sequences for all of the HLA genes, we can compare them to the database and find any exact differences down to single base pairs. In our populations, we found that about 1% of the cDNA sequences that were generated by StarPhase did not exactly match something in the IMGT HLA database. While these need to be verified, they could serve as candidate novel alleles that may be of interest to certain researchers. Lastly, we were able to look at novel rearrangements in the CYP2D6 gene. While we only found a handful of these across our entire population, they may be of interest to certain researchers who are studying CYP2D6, in particular how deletions and duplications get propagated within this complex gene. So, if you're interested in using StarPhase, the tool itself is publicly available on GitHub, and you can download it through Bioconda as well. It's currently built into our Whittle workflow, which is basically a workflow that will do alignment, variant calling, StarPhase, and various other tools that are common for HiFi sequencing. The only inputs you need for StarPhase are your reference genome, a phased VCF file, and an aligned BAM file, all of which are typically generated by most secondary processing pipelines. The tool is available at the GitHub URL posted at the bottom of this slide. And of course, if you need any help running StarPhase, feel free to reach out on GitHub and I'd be happy to assist you. So if you're someone who's interested in long-read HiFi sequencing or in pharmacogenomics, hopefully you found this information on StarPhase helpful. Thank you for listening and have a great day.